television programs.
Hello, teacher. Hello, students. Welcome to today's lesson on the multiplication of viruses. In our last lesson, we were introduced to viruses. We learned about how they are structured, what their four main characteristics are, and how they function. We also looked at how different viruses infect a cell. Today, we will learn how viruses multiply. We will compare the lytic and lysogenic multiplication of viruses. I hope you are ready to begin, students. Before we discuss how viruses multiply, we must begin by learning about bacteriophages. Most of the knowledge that has been acquired about virus multiplication has been done so through the work on bacteriophages, or phages. In particular, a bacteriophage called T4 has been the source of much study on the reproduction of viruses. Phages are viruses that infect bacteria. They multiply inside bacteria by making use of some or all of the host biosynthetic material. Depending on the kind of phage, it can exist in various forms, and the nucleic acid can be either DNA or RNA, but not both. There are many similarities between bacteriophages and animal cell viruses. That is why they are used to study the multiplication of viruses. Also, knowing the life cycle of bacteriophages is necessary to understand one of the mechanisms by which bacterial genes can be transferred from one bacterium to another. Phages may have a lytic cycle or a lysogenic cycle, and a few viruses are capable of carrying out both. In lytic phages, such as the T4 phage, bacterial cells are split open, or lysed, and destroyed immediately after the viron has replicated. As soon as the cell is destroyed, the new phages can find new hosts. In comparison, the lysogenic cycle does not result in immediate lysing of the host cell. The phages that are able to undergo lysogeny are known as temperate phages. The temperate phages gene will integrate with the host cell's DNA and replicate along with it. The virus remains dormant until conditions in the host cell weaken. Then the endogenous phages, also known as Prophages become active. At this point, the prophages initiate the reproductive cycle of the virus, resulting in lysis of the host cell. As the lysogenic cycle allows the host cell to continue to survive and reproduce, the virus is reproduced. We have covered quite a bit of material. Let us do an activity to ensure that we understand everything we have covered thus far. In the following sentences, fill in the blank with the correct term.
Students, let's get ready. Begin. Time's up! Let's get back to our lesson.
Welcome back, students. I hope you were able to complete these sentences. Let us review the answers together. The first sentence should read, information on virus multiplication has been acquired based on work with bacteriophages. The second sentence should read, phages are viruses that infect bacteria. The third sentence should read, phages may have lytic cycles or lysogenic cycles. The next sentence should read, when bacteria cells are lysed, it means they are split open. Finally, the last sentence should read, the phages that undergo lysogeny are known as temperate phages. Were you able to complete the activity? If so, great work. Let us look at the lytic and lysogenic cycles in more detail, starting with the reproductive cycle of the bacteriophage in the lytic cycle. The attacking bacteriophage, or virus, attaches itself to the host cell wall. The virus DNA is injected into the bacterium. Once in the bacterium, the viral DNA controls the production and multiplication of other virus DNA and protein coats. The DNA and protein coats assemble into new phages particles which burst the host cell. In the lysogenic cycle, the virus's DNA becomes integrated into the genetic makeup of the host cell. Every time the host cell divides, the phage's DNA is replicated. This means that every new cell will get a copy of the phage's DNA. A provirus is formed and replicated each time the host cell reproduces. After a while, the virus can start to assemble virus proteins. Once whole viruses are assembled, they leave the cell by lysis, or by ongoing exocytosis from the plasma membrane. Let us do an activity to review what we have just learned. Students, explain the 5 stages of the lytic cycle. You may begin now.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello again, everyone. I hope you were able to recall all of the steps. Let us review the answers. The first step is the bacteriophage attached to the cell wall of a bacterium. Next, the bacteriophage injects its DNA into the bacterium through a hole punctured by contraction of the tail sheath. In the third step, the virus DNA controls the production and development of more virus DNA and protein coats. Next, the DNA and protein coats assemble into virus particles. In the last step, host cell membrane is lysed and phages are released into the system to potentially infect other cells. I am sure everyone was able to recall the steps. Great work! We have reached the end of our lesson. Today, we learned about the multiplication of viruses. We discussed how bacteriophages have been used to gain knowledge on how viruses multiply. We also compared the lytic cycle and the lysogenic cycle. Until next time, thank you, teacher. Thank you, students.